All right, guys, you're getting caught by the recording now. I've rearranged the news items on our ICON website to remind you of the times that the TAs are scheduled to meet with you. You can also send them questions. And just to put it down in writing somewhere, I've written down which of the slides for Chapter 10 are not going to be addressed on the exam itself. So I hope that clarifies that. Okay, good point. Let's see, lighting. Thank you. People should correct me when I'm not paying attention. Oh, dear. Is the AV person still there? Can you get the spotlights off? Oh, dear. Oh, well. There we go. All right, so I just wanted to remind you of those things. Do people have questions about Chapter 10 and the carbohydrate metabolism that we covered on Monday and Wednesday? Any questions about that people want to ask? Okay. Well, let's move on to the citric acid cycle. And you are showing your wisdom by being here for the citric acid cycle, even though it's not formally on the test, because the things we're going to cover today and Monday are expansions on and will reinforce many of the concepts in the chapter on overview of metabolism. So you're going to have had a chance to see it in a new context. Citric acid cycle is a cycle that's had several names. I will try to stick with citric acid cycle, since that's what the text calls it. It's also been called the Krebs cycle and some other things. What's striking about the citric acid cycle, in addition to the fact that it's basically as central to metabolism as glycolysis is that the citric acid cycle is an amphibolic cycle. When we were looking at the pentose phosphate pathway, there were certainly aspects about that were amphibolic. That is, there were synthetic aspects, or under certain conditions you could use it to burn things. But the citric acid cycle is the one where the amphibolic character of it really comes to the forefront. And so it's going to, in some respects, or sometimes, or in some cells, be a catabolic pathway. It's going to break stuff down, and it's going to be oxidizing carbon atoms all the way down to carbon dioxide. And in doing so, it's going to be capturing metabolic energy in a form that will ultimately make ATP and maybe make a little bit of high energy compounds at the moment. Is there something that, okay, I just thought, okay. The second thing about the citric acid cycle is that it is a very versatile pathway. It has a capacity to burn all fuels. So you can pass sugars through the citric acid cycle, glucose, fructose, all of them. You can use it to burn fat. And we'll see that in later chapters. And even proteins if you get to the point in fasting where you need to burn your proteins for energy. It also has an anabolic face, a side where it's used to make new things or to make things for other purposes. We're going to see where it makes amino acids, which ones are likely to emerge directly out of the citric acid cycle, and we'll point sort of a roadmap towards others that eventually get out of it. It's very important for making the backbones for gluconeogenesis. And I want to emphasize again that gluconeogenesis needs both carbon backbones and it needs energy. And we'll get back to that, to remind you on that. And precursors for the heme molecules. And we've seen heme already in hemoglobin and myoglobin, but hemes are required for many proteins, especially the cytochromes. And so this will be the source for that. What also makes it a little challenging to think about is that not only is it a cyclic pathway that really goes around and around, it doesn't have, a starting compound and an ending compound. To make it even more interesting, it can be entered at any of several different places, and we'll be seeing that as we go along. So to set the context for this, you've seen this figure before about how biopolymers get made into monomers. Monomers get broken down into 2 and 3 carbon fragments. So far, the 2 and 3 carbon fragments that we've seen have come to us through glycolysis that pyruvates a 3 carbon intermediate. And we're going to see how you get pyruvate into the citric acid cycle. And then once those carbon intermediates, whether they're from sugars or amino acids or wherever, get into the citric acid cycle, it's going to have a couple of outputs. It's going to produce carbon dioxide. It's going to generate NADH, the reduced form, and QH2, the reduced form, which can provide reducing power to fuel oxidative phosphorylation. And this isn't really capturing very much of its biosynthetic aspects, but we will address that as well. So to get pyruvate specifically into the citric acid cycle, we have to get from pyruvate to acetyl-CoA. Acetyl-CoA is one of the common things that gets fed into the citric acid cycle from metabolism. It's the place where people most often think of the cycle as beginning, although we'll see that's something of a simplification. To turn pyruvate into something that can be used needs an enzyme called pyruvate dehydrogenase. And pyruvate dehydrogenase is, in most organisms, a large complex that is able to catalyze several distinct enzyme activities to accomplish the whole thing. Sometimes they're just simply called enzyme 1, 2, and 3. These complexes can have anywhere between 60 and 140 polypeptides. And this is just the E2 core, so this isn't even the whole of the pyruvate dehydrogenase complex. The reaction that's going to happen when all is said and done at the end of the day is pyruvate is going to be fed in, plain old CoA, coenzyme A, the empty form, NAD+, which is the oxidized form of the adenine nucleotide. And the products then will be acetyl-CoA, which you may remember has the same energy of hydrolysis as an ATP. It will release a carbon dioxide, and it will have produced a reduced molecule of NADH. So that's where we're going to get, but to get there takes a couple of steps. The first enzyme in the complex can be called pyruvate dehydrogenase itself, or, for less confusion, pyruvate decarboxylase. And what we're going to be doing in order to accomplish that is taking advantage of another cofactor. This is thiamine pyrophosphate here. And the thiamine pyrophosphate is one of the common cofactors derived from B vitamins again. And this ring here is the business end, if you like, the place where the action is going to happen. That hydrogen there in red is the specific point of activity. And so if we take a look at what's happening in here, here we have the thiazolium ring, and here we have pyruvate. And there's an attack, so that we now have a covalent bond to the thiazolium ring. That sets up this group to leave as a carbon dioxide molecule, and we have a covalent intermediate at this step of hydroxyethyl thiamine pyrophosphate. So we have now a 2-carbon fragment attached to the thiazolium ring. The second enzyme in the series, enzyme 2, is going to receive that hydroxyethyl group and oxidize it. And in the process, it will regenerate the first enzyme, so the first enzyme is free to pick up another pyruvate. So here is the hydroxyethyl group on the thiazolium ring. There's an attack on a lipoamide that's on the E2. And I guess I didn't introduce lipoic acid yet. Lipoic acid is yet another B vitamin. And it has this interesting structure up in here. This is the business end for lipoic acid. This sulfur is going to do the work. So what was a covalent attachment to the thiamine is turned into a covalent attachment to the lipoamide, and in the process it regenerates the E1's thiamine pyrophosphate. Furthermore, if you look at the structure of the carbon backbone at this point, so this is the hydroxyethyl group, and after the rearrangements at that stage, what you have here is now a carbonyl carbon. So this carbon has been oxidized. This is now in a relatively oxidized state compared to what it was back here. It's stuck onto the lipoamide covalently. So to get that enzyme back, we'll have to get it off somehow. And in the process to do that, we will have to transfer it somewhere and get some reducing power back to get the lipoamide back. The next step that's going to happen on enzyme 2 is that the lipoamide will be able to transfer that acetyl group onto the coenzyme A. So now you have acetyl-CoA or acetyl coenzyme A. Here's the acetyl group, the CoA. And several of the features of the acetyl-CoA that are important are it's freely diffusible. So now the acetyl group can leave the pyruvate dehydrogenase and be available for many other things. We're going to get it into the citric acid cycle shortly, but in fact it can be used for other things as well. Unfortunately, if you take a look down here, the lipoamide is in the reduced state right now. So the lipoamide is not able to receive another 2-carbon fragment until it can get oxidized. And so the third reaction is enzyme 3. Enzyme 3 will regenerate the lipoamide of enzyme 2, and the electrons that were in the lipoamide are transferred to another cofactor. We saw FAD in the overview of metabolism as one of the electron carriers. And so FAD will be able to receive the electrons at this point. Let me just get that actively there. FAD will receive the electrons and become FADH2. Now the lipoamide has been regenerated, and the enzyme-bound FADH2 can transfer the electrons to an NAD+, and NADH is now the reduced form, and it is free to diffuse away. Just to go back, I guess I won't, there we go. We did take a little bit of a look at this, I think, during the overview of metabolism, but if not, here we can see it in more detail. We have, again, an adenosine moiety here with a diphosphate linkage. There are so many of these cofactors that have an ADP somewhere in them, but this is one of them. And here is the side chain, and the business end in this case is the flavin ring that can undergo oxidations and reductions. This diagram is going to show them all fitting together, so you can sort of see the sense of it all at once. Here's your pyruvate, the 3-carbon molecule, oxidized at one end, reduced at the other. And in the first reaction, the thiamine is going to pick that up and attach it to the enzyme. So now we have the 2-carbon fragment on the enzyme, and that reaction is driven by the loss of the carbon dioxide. The hydroxyethyl moiety has to be oxidized, and that'll be accomplished with lipoamide. And now it's covalently attached to the lipoamide, but that frees up the thiamine for another round. The lipoamide can transfer that acetyl group to an incoming CoA to release acetyl-CoA. The reduced lipoamide has to be oxidized, which is going to be accomplished through an FAD that stays on the enzyme. And the electrons that go onto the FAD are transferred to an NAD+ to release NADH. So this is almost like an assembly line. This huge complex has the job of taking in stuff and putting out other stuff through a rather complicated reaction. And what you can see is that once the pyruvate enters the pyruvate dehydrogenase complex, it doesn't actually leave the complex until it's acetyl-CoA with the electrons and an NADH. There's some reason to think that it may provide for what's called channeling, so that all the intermediates are kind of funneled onto the right spot at the right time and not just sort of a chaos of collisions and so forth. 3 enzyme-bound cofactors, the thiamine, the lipoamide, and the FAD. 2 soluble cofactors, NAD plus the CoA, and the ability to make a freely soluble reactive acetyl group and a freely soluble set of electrons that can be used somewhere else. And this is showing you that in a little bit more detail about how it's built. In this particular case, the lipoamide reductase is in the center. You have additional polypeptides on there for the dihydrolipoyl dehydrogenase, and then sort of coating the outside, the pyruvate decarboxylase that catalyzes the first step in the whole reaction. Okay. So we've taken pyruvate, the 3-carbon fragment, out of glycolysis. We've used pyruvate dehydrogenase to produce one molecule of acetyl-CoA. So what's going to happen at that point? So this is sort of an overview of the citric acid cycle. We can see that acetyl-CoA is coming in at one point. We've highlighted a number of other things coming out of the citric acid cycle. So here's a carbon dioxide coming off and another one. So the 2 carbons that come into the citric acid cycle leave the citric acid cycle at some point. We also see molecules of NADH, QH2, NADH. So the carbons that are entering as acetyl-CoA are going to get oxidized at some point in the reactions of this cycle. And we can also see one molecule of GTP made down there. So this cycle as a whole is a way to oxidize carbon atoms. It's also a way to provide intermediates, and we're going to see sort of that function that's in tension with the catabolic or oxidative aspect of it. The first reaction is going to be 2 carbons added to oxyl acetate to make the 6-carbon citrate, which certainly looks like a symmetrical molecule. An enzyme aconitase is going to rearrange that to an asymmetrical isocitrate. The iso preface is just warning you that it's been rearranged a little. We're going to have a decarboxylation step at the isocitrate dehydrogenase that will produce reduced NADH. And now we have a 5-carbon backbone, 1, 2, 3, 4, 5. Alpha keto glutarate. Should sound close to glutamate, and we'll see why. Another oxidation reduction step with a loss of carbon dioxide. Now we have a 4-carbon backbone that's attached to a CoA backbone. So you have CoA as an input as well, succinyl-CoA. That 4-carbon backbone is going to then be released as succinate, and we'll see why that enzyme can drive the production of a GTP. There's another oxidation reduction reaction to a fumarate, still 4 carbons, addition of a water, 4 carbons, another oxidation reduction step, and we're back to oxyl acetate. Now we're going to look at those steps in more detail, but I wanted you to get a general sense of this pathway, that there's chain shortening in parts of it, there's oxidation reduction, and so on. Okay, so let's look at citrate synthase. It's named rationally. This enzyme is for making citrate. It's an essentially irreversible reaction, and that's because this thioester bond here, if you remember, has about the same energy as an ATP. And so it's like phosphorylating something. It's a way to drive the reaction in that direction. There's also quite a large change in conformation. You may remember how hexokinase closed like a clamshell. Well, you can see here citrate synthase sort of closing up from both sides. And what's it going to do? Well, here's oxyl acetate with the carbonyl carbon there. We're going to add this 2-carbon fragment to it, and by smushing it onto this carbon, we end up with this 6-carbon piece and a free CoA molecule. So this is actually a tricarboxylic acid. This is carboxylic acid, one there and one there. One of the older names for this cycle is the tricarboxylic acid cycle, because there are a couple of the compounds in it that are tricarboxylic acids. So if you ever see it, that's what they're talking about. The second reaction, the pathway is catalyzed by aconitase. Aconitase is not the most transparent name for an enzyme, but it gets that name because of the intermediate which is a connotate that looks like this. And what a connotate can do is take out a water molecule and add one back, and it just scrambles the position of that hydroxyl group from this middle position here to one that's staggered off the center line. And so you can imagine easily that that would be a freely reversible reaction. And this compound, citrate, is itself a symmetrical compound. And yet by using radioactive carbon markers carefully, we can show that it always occurs, this reaction always occurs in one particular direction, which seems contradictory. But we're going to see in a little bit that why that's true, and that that was one of the clues to how enzymes do what they do. So the third step is isocitrate dehydrogenase. So this is isocitrate with the alcohol down one. It's a dehydrogenase, so we should expect to see some reduced cofactor produced, which we do. NADH is the reduced cofactor. And this formation of the NADH is driven by a decarboxylation. And this is just showing you that the oxidation reduction step happens before the decarboxylation, but that's not necessarily terrifically important for you unless you're interested in enzyme mechanisms. The loss of that carbon dioxide helps make this reaction irreversible. So this is one of the one-way steps in the citric acid cycle. And again, the 5-carbon product of this, alpha-ketoglutarate. And glutarate is an organic acid related to glutamate, but not the same thing exactly. Fourth step is alpha-ketoglutarate dehydrogenase. Should warn us something's going to get reduced. It's a second oxidative decarboxylation. It's another step where NAD plus is going to be reduced to NADH. But in this case, we're going to actually transfer the new 4-carbon fragment onto a CoA molecule. So here you have the alpha-ketoglutarate. It's a dehydrogenase, so you can see we've produced NADH here. And this is the carbon that is going to be the carbon that's been reduced. And so as a consequence, we have here the succinyl-CoA 4-carbon fragment attached to the CoA molecule, and that's driven, that reaction is driven by the decarboxylation. Now, if that looks a little bit familiar, it should. We're putting in this organic compound that up to here about looks a lot like pyruvate. Our soluble inputs are CoA and NAD+. Our products are carbon dioxide and NADH and a CoA compound. All of that sounds a lot like pyruvate dehydrogenase. Very, very similar overall look to it, and it turns out that alpha-ketoglutarate dehydrogenase is actually a multi-enzyme complex with many similarities to pyruvate dehydrogenase. So when you learn one, you're halfway to learning the other. Or it's another example of how a good metabolic strategy or a good catalytic strategy is usually used more than once in metabolism. Any questions? Okay. So we've seen 2 decarboxylation steps so far, and it's reasonable to ask which carbons are going away. This is a little subtle. It turns out that on the first cycle through, the carbons that just came in on the acetyl-CoA do not get lost. They stay with the carbon backbone. And let's look at that schematically here. So the steps we've looked at so far are citrate synthase, where a 4-carbon fragment picks up the 2 carbons from acetyl-CoA, and now we have citrate. And they've color-coded this. Is that visible as different at all on your handouts? Good, good. So now we have the 6-carbon citrate, and we have a connotate, which you might think would scramble it, but it turns out it doesn't. And we can show that schematically by keeping those top 2 as red. Alpha-ketoglutarate at this point nibbles off the carbon dioxide from the middle, and the succinyl-CoA nibbles off one more. But the 2 carbons that entered with the acetyl-CoA are still in there. We're going to see that succinate and the subsequent steps do allow for scrambling. And so when we get back to oxaloacetate, it's going to start to be scrambled. And the next pass, or somewhere after that, the carbons that came in as acetyl-CoA will leave as carbon dioxide. But on the first cycle of the pathway, they don't. That's a little strange. I'll need to stop and see if anybody has questions about that. Okay. So we've made a succinyl-CoA. Succinyl-CoA, like acetyl-CoA, like CoA compounds in general, has a high-energy bond attached to the CoA molecule. And the next enzyme in the cycle is called succinyl-CoA synthetase. It's named for the backwards reaction. Okay? What it does in the context of the citric acid cycle is we are going to end up making a free succinate and a GTP and releasing the CoA completely. Some of these steps in here are the intermediates along the way, but they're not the products of the reaction altogether. This is showing you that kind of. This is showing you at the. Let me see if I can back up one. So in the first reaction of the step, a phosphate. So this is an inorganic phosphate at this point. Does not have any connections to carbon. It's just phosphate ion. Is going to come in and replace, if you like, the CoA molecule. So you have a succinyl phosphate, and this bond here is going to be of comparable energy there because of the structure of the molecule. Ultimately, that phosphate will be donated to a GDP, so this will be an example of substrate-level phosphorylation. Where else have we seen substrate-level phosphorylation? A pathway? Glycolysis? Okay. Don't scare me too much, folks. And so, interestingly, that succinyl phosphate can transfer its phosphate to a histidine to sort of park it as a temporary high-energy intermediate. That allows the succinate to go away as a free compound now. And this is now just a symmetrical 4-carbon backbone with 2 carboxylic acids. That phosphohistidine is capable of transferring its phosphate to a GDP, and that's when we get the GTP out of the compound. So this is substrate-level phosphorylation. We can make a fresh high-energy bond from something that did not have one to begin with. And again, just to make the point again that it's named for the backwards reaction, that it's starting from succinyl-CoA in the cycle. It's named backwards. So now we have some succinate. Here's our succinate, 4-carbon fragment with 2 carboxylic acids, one at each end. The point of the next few reactions are several-fold. We're going to regenerate oxaloacetate so we can keep going as a cycle. And we're going to get a little bit of oxidation reduction action happening as well. So the next enzyme is succinate dehydrogenase. Here's your succinate. Once it's been dehydrogenated, we now have a double bond. So we've created an unsaturated bond. And something had to have gotten reduced if this is now a double bond. And here's your FADH2. FAD received the electrons, became FADH2. And interestingly, this enzyme, succinate dehydrogenase, is actually a mitochondrial membrane enzyme. So we haven't talked about the location of the cycle as a whole yet. It's in the mitochondria. And this enzyme, and only this enzyme, is actually a membrane protein. And the electrons that are on the FADH2 are then going to be transferred to ubiquinone to make reduced ubiquinone. So we're getting a chance to see several of these electron carriers in their context where they're actually doing something useful. These reactions are also ones that we're going to see again in other pathways. So they're worth paying attention to for that reason, that we're going to go from this fairly reduced central core to something that's partially oxidized. Now we have fumarase. One substrate is fumarate, which we just made from the succinate dehydrogenase. And fumarase can catalyze a reversible reaction. This is just adding water across that double bond. So a freely reversible one. So you have a double bond there. If we add water across it, we now have an alcohol over there and a single bond there instead of a double bond. This is a freely reversible reaction, and this kind of behavior at the level of the carbon backbone we will see again. So it's fumarate, gives rise to malate. And now we're back to where we started from. The eighth and last reaction in the pathway, malate dehydrogenase. Again, something's going to end up reduced. It's going to oxidize the malate and reduce NAD+. Here's the NADH in the reduced form. And here is where we're seeing the oxidation happening. This carbon had one bond to a hydrogen there and one to an oxygen. At the end of this, it's got two bonds to the oxygen. So this is the carbon that has been oxidized. That regenerates the oxaloacetate. There's no carbon dioxide coming off, is there? So this isn't a very favorable reaction. There's nothing on the face of it that looks like it should make it go that way. And in fact, the standard free energy change for it is plus 30 kilojoules. So you would not expect it to go in that direction at any significant level at standard conditions. However, in the cell, we're not at standard conditions. And in the cell, the oxaloacetate is removed by the irreversible reaction at citrate synthase. And so although this is not favored in that direction, the very large free energy change associated with citrate synthase pulls it forward. Other questions? Okay. All right. I'm not quite sure why I included that slide. Let's come back to that later if we have time later, okay? Oh, that can't be right. Excuse me. Can that be right? Oh, okay. That's not right. I'm missing part of my slides. Let me go back and see if I can find this. No, okay. That was a shock. Yes, I must have somehow or other, or possibly Microsoft somehow slipped on me. There we go. There we go. Okay. That was a shock. Okay. So let's stand back a little bit and look at what the citric acid does for us energetically, why it's such a powerful part of the core of metabolism. We looked at glycolysis, and glycolysis as an anaerobic pathway doesn't produce very much energy out of a glucose molecule. One glucose molecule will give you 2 ATP under anaerobic conditions. It's only showing more ATP coming off if you can regenerate that NADH in a way that's going to produce ATP. If you're under anaerobic conditions, you regenerate that NADH by making alcohol or lactic acid or something like that. So glycolysis alone is not very good at getting most of the energy out of glucose by oxidizing it. However, the two pyruvates that glucose provides in one pass through glycolysis start to produce a lot more energy when we put it in the context of the citric acid cycle. So pyruvate dehydrogenase will produce an NADH for each pyruvate. Since we've got two pyruvates, two NADH. We're going to see when we talk about oxidative phosphorylation in more detail that each NADH produces about 6 ATP. So we're starting to look at the increase in yield under aerobic conditions or when oxygen is available. So those two pyruvates produce two acetyl-CoA molecules, and this is really where the big yield comes from in trying to oxidize a glucose molecule. So each of those acetyl-CoAs would make 3 NADHs, 6 for the whole glucose molecule. That's 18 ATPs from the dehydrogenases that are making NADH. We're going to get 2 molecules of QH2. We're going to see those don't make quite as much ATP, but that's 4 more ATP. And we had 2 molecules of GTP made by the substrate level phosphorylation. And if we wanted to count the carbons, we lost 2 carbons here as carbon dioxide, and this is 2 times 2 carbon dioxides, so the other 4 come out in the context of the citric acid cycle. This is a powerful way to extract energy from the glucose carbons that were in the backbone, or to oxidize it. But the reactions that we use for oxidizing pyruvate that came from glucose are equally powerful at extracting energy from amino acids or from fatty acids. So this is a very powerful catabolic pathway in that respect. Moreover, one of the advantages of a cyclic pathway as opposed to a linear pathway is that if you increase key intermediates, you can increase the flux through that cycle disproportionately because each one can be used multiple times. And so you can partly control the activity of the citric acid cycle by adding more intermediates into it that will just get used over and over again to keep oxidizing carbon backbones. When we add this all up, one glucose molecule that is oxidized completely aerobically can give you about 38 molecules of ATP. Contrast that with about 2 in the absence of oxygen. So clearly, the first organism that got a citric acid cycle going well had an immense advantage in terms of extracting energy. Now, there is also a regulation of the citric acid cycle. And as you might expect, key regulation is going to happen at irreversible steps. It doesn't make sense in the case of the citric acid cycle to talk about committed steps because it's a cycle. It's not a linear thing, it's a cycle. And so there isn't really a committed step in the usual sense that we think of for a linear pathway. But there are irreversible steps. And let's review which are the 3 irreversible steps. So citrate synthase is an irreversible step driven by the energy of that acetyl-CoA thioester bond. Isocitrate dehydrogenase to alpha-ketoglutarate is an irreversible step driven by a decarboxylation. And alpha-ketoglutarate dehydrogenase is another irreversible step, again, driven by a decarboxylation. So these are going to be good targets for regulation. And the kind of things that will regulate their activity make very good sense in terms of the catabolic purpose of citric acid cycle. So if we look at citrate synthase to begin with, citrate itself will inhibit the activity of citrate synthase, so it's a direct feedback. It will also be inhibited, let me see, it'll be inhibited by NADH. Now, citrate synthase doesn't make NADH itself, but that's one of the overall purposes of the pathway catabolically, and so that's an indirect feedback inhibition. The NADH being abundant is a sign that the energy has adequate energy. And the other that is sort of a little hard to follow here is that succinyl-CoA can come back and inhibit citrate synthase. Well, succinyl-CoA is at the end of the irreversible steps, so if it's accumulating, it makes sense to inhibit these steps in there, and it's another regulatory factor. Here's isocitrate dehydrogenase. Again, it's inhibited by NADH. That would be a direct feedback inhibition. It's also reading the overall status through the concentration of NADH. Positive regulators are ADP. We've seen before that ADP is an indicator of relatively low energy availability. So if you have an accumulation of ADP, maybe it's a good time to turn up the flux through the citric acid cycle. Calcium is a regulator in the context of the mitochondria and is something that's controlled outside the cycle itself. Here's the alpha-ketoglutarate dehydrogenase. Again, NADH feedback. We have the direct feedback through the succinyl-CoA again, and the stimulation by the calcium there. So these three steps for regulation are irreversible steps and therefore logical ones to be hitting. And all these regulators, with the exception of calcium, are things that make sense in terms of the energy status of the cell. Any questions? Okay. I think rather than trying to do the evolution of the citric acid cycle in just a few minutes, I'll call that a day. I'm happy to stay here and answer questions if people want to ask questions about the rest of the lecture or about carbohydrate metabolism. Lynn, do you have a review session scheduled? Okay, before you leave, please, Lynn will have a review session on Monday at 5 o'clock in, okay, over in Bowen, and she'll put a notice on Icon about the room and location. You sent an email, okay, okay. Yeah?
I was wondering if sometime you have some time to talk about the class and maybe...
Sure, let me just turn this off.